Good morning, everyone. So I'm going to present a little bit of our very preliminary results of what we have been studying so far regarding PRRS diversity. And so molecular epidemiology of PRRS has become a very popular approach in the field, especially looking into outbreak investigations. It has the potential of identifying viral introductions in a farm, but also identifying viral persistence in farms undergoing elimination. But it can also be a useful tool to guide decision-making regarding PRRS control in the farms. So, for example, we would expect to have some homologous, some protection if we have homologous infections, and some variable cross-protections between heterologous infections. However, what defines homologous and heterologous infections is still not very clearly defined. But regarding genetic diversification of the PRRS virus per se, I'm going to give you a few examples here regarding PRRS specifically. So it could be genetic diversification can be either through mutant generation, which would be in PRRS a mutation rate estimated at 4.7 to 9.8 times 10 to the minus 2 substitutions per site per year. And the other form of genetic diversification could be also recombination. And with that, we have a recently published paper as an example of recombination in PRRS. And this is specific isolate of PRRS type 1 from Europe. If we look only at R5, we could categorize this virus as a field virus or a wild-type virus. However, if we look at R6 or R7, we would classify this virus as a vaccine-like virus. Furthermore, since PRRS is an RNA virus, we also have the concept of Quasispecies, which basically means that since RNA viruses are usually very prone to mutations, we likely have a very heterogeneous viral population either within the same individual, which in this case would mean a pig, but we also would expect to have a very heterogeneous population, viral population within different organs or even within a cell. So as we see here in the picture, if the mutations are random and the population is in equilibrium, we wouldn't see a change in the consensus genome. However, if something happens and a mutation becomes more prevalent, then we would see a change in the consensus genome. Regarding PRRS viral diversity per se, we know that PRRS type 1 and PRRS type 2 have around 40% genetic difference between both of them. And regarding PRRS type 2 specifically, we can even subclassify this virus into lineages, which should have around 10 to 15% nucleotide difference between each lineage. But furthermore than that, this paper from Poplowski and collaborators recently published actually showed us that through time we actually have a very dynamic turnover of lineages over time. So we can see here that they showed that some lineages were actually extinct over time, over this period of time, and some sublineages actually emerged during this period. So it is a very diverse and heterogeneous population. So what do we know about PRRS diversity in growing pigs? Not much really, but we could speculate that we could expect some viral diversity to be higher in this population, and this could be because first, they have a lower biosecurity usually than in sow farms, which are most of our knowledge so far, and this could potentially increase their vulnerability to new viral introductions. Also, it's not unusual that animals from different sources are commingled, so with that, we could expect to have maybe more than one virus circulating, and this could possibly lead to recombination or maybe reinfections. We don't know many of those things. Also, the passive immunity in young piglets is of short duration, so by the time passive immunity weans out, they might become susceptible to new infections or reinfections. And also this is a population in which vaccination is also not unusual. So again, we have the same caveats of introducing another live virus in this population. So regarding that, we decided to take a look at our data that we have. So I'm working with AIM-SHIMP data, the Morrison Swine Health Monitoring Program, which is a national-wide monitoring program for endemic swine diseases, PRRS being one of the main ones. We currently enroll about 50% of the swine population in the US, and all of those systems that participate in our monitoring program voluntarily share all of their R5 PRRS sequences with us from their routine monitoring. So far we have over 30,000 sequences. So with that very large data set, we try to take a look a little bit on PRRS diversity on that. So what I did here was I calculated the percent nucleotide difference from each sequence to the reference VR2332, which is a historical reference. And we can see here that in breeding groups, the range in difference that we found was much narrower than what we found in the nursery, finisher, or win-to-finish sites. So that indicates that perhaps we do have a more diverse viral population in those types of sites. However, what about diversity within farms? So within one farm over a different period of time, within individuals from the same farms, or even within the same individual over time. So in this case, we want to look, as we were before, we are looking only at the consensus genome, and we want to see if we can capture that change in the consensus genome. So again, we looked at the AIM-SHIMP data set, and we selected farms that had at least 5 samplings from the same farm with up to 30 days apart from each collection point. So this is an example of one farm, and that's more or less what we would expect to see. We see a few point mutations here and there, but overall the virus seems to be stable and fairly similar to each other, to the first ones and to each other over time. However, it was not uncommon that we saw situations like this, in which we have, for the first 4 samplings, one virus identified, which was fairly similar to each other. But then by day 42, we have a different virus identified. And that virus stayed in the farm until week 90, when then the first virus appeared again. So since we're only talking about 3 months difference, what we believe might be happening is a situation like this, in which the farm is probably receiving animals with different virus originally, and since we only have one sample from the farm in one point in time, we sometimes sample virus from farm A and sometimes sample virus from farm B. So it's very hard to try to understand viral evolution in a farm in a scenario like this, because we might not be capturing everything, all of the diversity within that farm with just one sampling. So then again, we looked at farms that had more than one sampling per collection time, and again with less than 30 days apart between each collection time. And what we saw is a situation more like we would expect regarding the case that I described above. So just visually here, I separated the virus into virus A and virus B. And we can see that for example, on day 0, it's clearly that the farm has at least 2 different viruses circulating. However, on day 11, only one virus showed up. On day 18, both viruses showed up, and so on and so forth. So if we compare, if you try to look at the evolution of just virus B, we would see that from the day 0 and the last day that they sampled, day 55, we don't have a lot of differences. However, if we look at day 0 and day 18, and day 18 to the other days as well, we see that the virus is quite different. So maybe that farm doesn't have only 2 viruses circulating, it could possibly have 3 viruses circulating. And that sampling, at least that first sampling, wasn't enough to capture all of that diversity as well. And then if you look at virus A, which had multiple samplings within a same time point, we can see that even within the same time frame, we have a lot of diversity as well. So the virus was rarely 100% identical. But overall, this particular virus was perhaps a little bit more stable, and we didn't see that many differences during that period. So just out of curiosity, percent nucleotide identity between virus A and virus B was around 87%, and virus A was similar to VR2332. So we wanted to further investigate that case, so we started a cohort of piglets from one farrow-to-wean farm that was undergoing a PRRS outbreak. So this farm was from the Midwest, and they reported the virus on May 21st, and that viral detection was actually after more than 2 years with a naive herd, so all of the animals were seronegative. So once they detected that virus, which was RFLP134, they performed LVI 9 days after the viral detection. So all sows and gilts were inoculated with the recently introduced virus. And we went in that farm 2 weeks after LVI was performed, and we sampled all of the animals from all 3 to 5 days old litters at that farm at that point in time. And we ear-tagged the piglets so we could follow them individually for up to 17 weeks. So our sampling was on 3 to 5 days old, before weaning, about 1 month into nursery, before moving them to the finisher, and then about 1 month in the finisher. So here, not really related to PRRS diversity, but for this particular population, mortality was very high. We started with 127 animals, and by the end we had only 22 survivals. CT values were high in the beginning, and they remained positive until our second sampling at the nursery, but by the time of our sampling at the finisher site, they were all negative. So if we look at the diversity and evolution of the consensus sequences for this population, we are here just, again, a preliminary result, showing a preliminary results of R5 sequencing of a few randomly selected animals. So we are looking at the first sampling at the farrowing room at 3 to 5 days, and then at the first sampling at the nursery, which was 6 weeks after. So on the farrowing, on the first sampling at farrowing, we had, again, not all viruses were completely identical regarding the consensus genome. We had 99.7 to 100% identity. At the nursery, the same thing, although diversity increased a little bit. And between both sampling, we had 99.5 to 100% identity of all of those viruses. However, the interesting thing is that now we can look at the evolution within each individual animal. So we want to compare the virus from, for example, animal A in the farrowing with animal A in the nursery. And with that, we see that most of them had some mutations occurring during that 6 weeks period. And that variation ranged from 100% to 99.7%, which would be equivalent to 1 to 2 mutations during that time. And interesting, just out of curiosity, one of these animals had a mutation in a particular site, which actually made the RFLP pattern change. So that's pretty much it that I have as a preliminary result so far. But I'm afraid we end up this talk with actually more questions than answers. So we saw that the range of percent nucleotide identity compared to VR2332 in growing pigs seems to be higher than among breeding herds, and that might indicate a higher diversity. However, we also saw that it's hard to estimate diversity over time within a farm with only one sample in each point in time. So if this is something that we really want to pursue, we should maybe perhaps reconsider how we are sampling those animals for that particular purpose. And although with limited power, we observed that R5 mutations ranging from 0 to 2 nucleotides within individual pigs in as little as 6 weeks. All of those mutations were non-synonymous, and each one of those occurred in different sites in the R5 gene. So as a next steps, we are moving forward with that, and we want to look now at diversity and evolution of PRRS variants, which would mean we're going to try to look now at the quasi-species level. So we want to see how that population cloud, viral cloud, changes over time within each animal and within the farm. So that's pretty much it, and I want to just thank our sponsors and our partners and collaborators for that, and thank you.
Meanwhile, we get the microphone there. In the next step, are you also going to look at the whole genome?
Yeah, so we are doing, we are doing the Minion sequencing, which Declan will show you a presentation tomorrow about the pros and cons of the technology. And we're not sure if we're going to be able to, we're probably not going to be able to get the complete, like 100% coverage of the genome, but we're going to be able to have at least the most important parts of the genomes, and we hope to get a very good result with that.
That's nice talk. In addition to the general mutation, so have you observed some no cinemas or cinemas variants in some coding region for no structure protein or in structure protein? You may mention that, but I didn't follow.
Sorry, can you repeat that?
Yeah, I mean, have you observed some enrichment for the mutation in some coding region for no structure protein or structure proteins?
Yeah, so since we only had 1 to 2 point mutation at the time, we only looked at the change in amino acid at this time, but we hope to, once we get to the next step, which is the identification of variants within each sample, I think we should be able to see if there are some specific changes in specific parts of the genome, if that answers your question. Sorry. No, we don't know yet, yeah.
Any other questions? No, with that, thank you very much, Mariana.